So if you're listening to them, I'll try to repeat the question because you guys aren't mic'd. I guess I can pass around the mic so that you can hear it on the recording. If I make a mistake, it's for all the world to see, but it's just a live recording so that you guys can use it. It's not like we're going to polish it up and podcast this thing. Okay. A little bit of review from what we covered on Monday, talking about pathology, right? So it literally translates to the study of suffering, like we described, which is kind of morbid in a way, kind of funny in a way. But the thing about pathology that I really like, I'm a scientist, I'm not a clinician, but I'm a very applied scientist. And what that means is I like to work on things that actually have a direct impact into patient care. It's very rewarding, in my opinion. I love the discovery part. I love understanding mechanism. I like this discovery for the sake of discovery. But to me, it means a little bit more when you hear that people are feeling better, or you've saved a life, or the quality of life is improved. So I'm a very translational scientist. And so pathology kind of bridges clinical practice and basic science. And I think that's one of the reasons I'm drawn to it so much, is we talk a lot in this semester about the who cares, like what does this mean for the patient? Okay, so there's a lot of discovery and a lot of really cool topics and understanding, but a lot of times it relates to something that's very clinically applicable. It investigates the causes, or we said etiologies of disease, and those underlying mechanisms, just like we did with Barrett esophagus. So if you think through the semester and you want to have a little outline of how to consider different diseases that we come up against, just think back to this first example of Barrett and walk through kind of the 4 steps. And I think it'll help you study with a little bit of a framework. And Barrett is a great example. We'll kind of keep pointing back to this example because it's the first one we talked about, right? A lot of times you remember like the first thing that somebody says, and then maybe you remember like the last thing. So Barrett will probably stay with you the entire semester. All right, so let's talk about cell injury. Like in Barrett, what's really going on at the level of that stem cell or that progenitor cell that's causing it to change? What's that injurious stimulus doing? And we're going to look all semester at different types of injury that's happening to the cells, and then how the cells respond. And now you know exactly how the cell responds with Barrett to lead to metaplasia, which leads to esophageal adenocarcinoma, which then would go on to form cancer. Okay? So we're going to talk a little bit more about the intracellular things that are happening and at the tissue level. So, in our environment every day, we're under attack. Our cells are being challenged. They're being tasked with the ability to respond to different types of challenges. So, for example, if you go outside in Flagstaff, it's typically sunny, except I'm very happy that it rained a little bit this afternoon. Okay, so UV radiation here is actually very intense. And there is a response that happens at the cellular level to radiation. And of course, our bodies are designed, many of us, our bodies are designed. I see a few of you who aren't designed as well to combat UV radiation because your skin is maybe a little bit lighter than others, right? So we have what protein that's manufactured at the skin to block radiation? What is it? Melanin, right? Not melatonin. That comes out of the pineal gland and makes you sleepy. Different. Melanin, right? That's the brown pigment that's manufactured from melanocytes, and that's what gives us a coloration or brown in our skin. That's why you tan, okay? Some of you skip that step and you just go from fair to red, okay? I understand. But that is a very important adaptation because radiation is harmful to DNA. And if that radiation disrupts the DNA in the cell, it's going to mutate. It's going to go through a change. Okay, so we saw that acid, a chemical, in Barrett, can cause a cellular adaptation. Well, radiation is another example that can cause a cellular adaptation. Okay, and what kind of cancer are you at risk of if you spend too much time out in the sun and you get that type of UV radiation on a constant basis. Just skin cancer, right? It could be, yeah, some of you want to say melanoma, some of you want to say, you know, squamous cell carcinoma, right? All sorts of, there's basal cell carcinoma, but skin cancer is the broad category. So under a normal condition, our cells see stress, whether that's chemical injury, or whether that's radiation, whether that's a pollutant, okay? If you live in a big city, right, if you look at your iPhone, you can see the air quality index is probably good here in Flag, and usually always is good unless we have a fire like we did, you know, like a month ago. But if you live in a big city, like in Phoenix, they'll have poor air quality indices that'll show up. Or LA, right? A lot of smog areas will have that. So what's going on there? Well, there's a lot of pollutants that are targeting not necessarily the skin so much or the esophagus, but what tissue is at risk of those harmful stimuli or stress. Lung epithelium, the bronchioles, exactly. Okay, so we're constantly being bombarded with stress. Well, we have the ability to adapt, and we can battle that, right? We can release more melanin and darken the skin, right? We can actually send in macrophages to gobble up the pollutants at the level of the lung tissue. But at some point, if it's chronic, like what we saw with Barrett, there's actually cell injury that is permanent. It's not able to adapt. And then if it progresses, it moves on to have what might be considered reversible cell injury and subcellular alterations, but there's some point of no return. And we're going to look at that. There's a point at which the cell just says, it's not worth trying to save this thing, we're just going to let the cell die. So now we're going to start talking about cellular death. And there's two types of cell death. There's what we refer to as necrosis, and I alluded to this term in our slide on peptic ulcer in the last lecture. And I said, oh, the tissue looks like it's a little necrotic. What does that word mean? What does necrosis mean? It is not programmed cell death, but that is the correct answer for apoptosis. Okay? Programmed cell death is apoptosis. So you can have credit for that one. But what does necrosis mean in the back? What's it? I can't hear you, sorry. Decaying. It's a tissue type of decay. Some idiot put a fan up here, so I can't hear anything. That idiot was me, for the record. Decaying. So it's an unorganized decay of the tissue. That's necrosis. Okay? It's like there's no organization. You're just going to walk away and it's going to fall apart. That's necrosis. So apoptosis or programmed cell death, some of you be like, why would you want to kill cells? Does that cross anybody's mind? So in development, it's very important because apoptosis is what, everybody hold your hand up. Apoptosis is what allows the tissue in between your digits to go away so that you actually have well-described and delineated digits. Right, does anybody know anybody where that didn't happen all the way? Maybe in the feet, like webbed toes. A couple friends might have some. Yeah, I have a friend or two that has webbed toes. So that's apoptosis that allows for cell death to take place in utero, where you get inner digit definition. Okay. Now let's look at physiologic versus pathogenic. So physiologic adaptations versus pathologic. So physiologic adaptation, so there's a lot of white space, you should probably write these things down. Physiologic adaptation is a result of like a normal process, such as embryonic development. So the cells are going to change, like apoptosis in between the fingers or the toes. Pathogenic cellular adaptation is the result from a disease, or where you've got a disturbance of the system, like what we saw with Barrett. This is a perfect example of a pathologic or a pathogenic adaptation. Now the cells can go through what we call atrophy, where they shrink in size. cell shrinkage is atrophy, or hypertrophy, which is an increase in cell size, hypertrophy or hypertrophy. And in some cases, they might increase in cell number, which is hyperplasia. That's the fourth term on the sheet here. And then we already described cellular metaplasia, which is from the Greek to change form. So all of these things are adaptations that can happen to a cell, a single cell. So let's use a different example than the GI tract. Let's look at cardiovascular. Now we'll come back to cardiovascular at the very end of the semester, but what I want to show you today is that these adaptations are happening throughout the body and throughout our lifetime in different systems all throughout the body. So this is a myocyte. The myocyte is really the workhorse cell of the heart. This is the functional cardiac myocardium. And it boils down to an individual myocyte. If you remember back to 201 when you're characterizing tissues, bless you, speaking of tissues, we have striated muscle and we have non-striated muscle, right? So smooth muscle is non-striated, and cardiac and skeletal are both striated. They're striated because the sarcomeres are organized in a very specific fashion, where you have the actin and myosin that overlap and they alternate. You guys remember that? Well, this is also branched, unlike skeletal muscle. Skeletal muscle is all in a nice organized parallel line, and cardiac is actually branched. But this cell can have an injury where you restrict blood flow. It's a cell that needs oxygen to survive. And so if a patient has a blockage of a vessel that perfuses the heart, they have a heart attack, what's called a myocardial infarction, and we'll talk about that at the very end of the semester, but just foreshadowing of what's coming. When you restrict blood flow to the working muscle of the heart, then those cells are at risk of cellular adaptation, right? If you rescue it fast enough, you can get them to the ER, they can get into the cath lab, they can put a stent in, reestablish blood flow, then they might actually go back to normal. If they don't, then they will lead to a scar, or what's called a fibrotic lesion. Okay? Not much different than a scar like on your skin. And we'll talk about later in wound healing, we'll talk about scarring or fibrotic lesions. But just remember now, you've heard the term, oh, grandpa had a heart attack, but phew, good news, there wasn't a whole lot of scar tissue. Like, what the heck are they talking about? Well, that means that they caught it early enough and they were able to rescue the cell before necrosis or decay set in. Okay? So here's the cell injury adaptation here as another example, other than reduced blood flow. This one is a load. So let's say that the individual has high blood pressure, they're hypertensive. Now the heart has to work against high blood pressure. Okay? So the heart has to have a pressure to open the aortic valve that's higher than mean arterial pressure in the aorta, otherwise the valve doesn't open. So if your blood pressure is high, the heart has to work harder to open the valve. That's why high blood pressure is a problem. Okay, so if you have high blood pressure and the heart is still working, it's working against the load. And what do we know when we load skeletal muscle? If you load it with force over time, what does it do? It hypertrophies, it grows in size, right? That's why people go to the gym. Now, the same thing happens to the heart, and so now you have a hypertrophic myocyte, and you have an enlarged heart. If you take the load away, a lot of times these individuals revert back. If you lower blood pressure, their heart will go from an enlarged condition back more to a normal size. Okay, so that's an enlarged or a hypertrophic heart. Well, in some circumstances, like back to the blood vessel analogy, if you don't make it quick enough, Grandpa didn't get to the cath lab quick enough, okay, they survived, but it might be that you lost some of these myocytes, and that was cell death, it was necrosis, and so now here you can see kind of this yellowish color. This is the scar tissue inside the heart. That's the fibrotic, non-reversible damage. Make sense? Well, here's another example. Instead of hypertrophy, let's talk about atrophy. So these are two different individuals, and A is the normal brain of a young patient in their 20s. Okay, obviously at autopsy. Okay, and on the right side, or B, is an 82-year-old at autopsy. And they passed away from non-brain related reasons. Okay? So what we're doing is, and they're approximately the same size or mass. They're just different ages for the most part. But what I want you to, what are some observations that you know from the young brain versus the old brain? And the meningeal layer on the brain's left side, this is the patient's left, you can see the meninges are still intact on the patient's right side. So the left side of the meninges in both young and old have been removed, so you can appreciate certain architecture. So I want you to tell me what you see. Deeper crevices, right? More exaggerated sulci. You see that? You see the big crevices of the depth? So we have a reduction in mass of the brain as we age. And this is because as we age, we have narrowing of the blood vessels that happens all over the body, including the blood vessels that perfuse the brain tissue. And so slowly over time, as we age, we're getting slightly less blood flow to the brain. And that tissue responds by atrophy. Okay, so just non-Alzheimer, right, non-neurological challenged individuals as we get older have more difficult time with brain function. They forget things, they can't remember names of stuff, and it's not like they've got a neurological disease. What they have is they have age. Age is a pathology. This is a perfect example, okay? So, there's no traumatic brain injury. The only thing that's happening between these two patients is age. About 50 years difference, and that's what happens as we get older. Tissues do atrophy because of reduction of blood flow, okay? Hypertrophy. Oh, sorry.
Did they also reduce in size from the synaptic pruning? Is there also a reduction in size due to synaptic pruning?
Yeah, you have both going on. So synaptic pruning, like as we age, we do have death of neurons that take place. And you don't regenerate new neurons in the central nervous system. So unlike the skin or the integument, there's not like a turnover. There's not a stem progenitor layer for new central nervous system neurons. So yeah, you do lose neurons as you get older as well. So that's part of it. You have the combo. The numbers that you're seeing, the neuron as far as mass is so microscopic. On a gross view, what you're really appreciating is the reduction in blood flow versus if you did a microscopic view, you'd be able to count. You would see differences microscopically on the point that you're making with synaptic pruning that takes place. Okay, so this is hypertrophy, an increase in cell size, and this is physiologic. This is not a disease. So on the left image, you've got a gravid uterus, the big one, or a pregnant uterus, and then you have a normal sized uterus down here. Okay? So here's our pregnant uterus, and this is a normal uterus, or the word here is gravid uterus. So this is hypertrophy, an increase in cell size that's physiologic. Right? This is not a pathology. Pregnancy is not a pathology, although many mothers would argue with me. Right? Trust me, it seemed like a disease at the time. But it's a normal process that's supposed to happen. And so you can see the increase in gross tissue size. And then if you look microscopically, this is the smooth muscle. Smooth muscle are like these spindle cells. So they kind of look kind of fat on one end, right? And then there's a nucleus here. So that's what a smooth muscle cell is going to look like. So if you look at the middle image, you can find, again, hematoxylin, and you can find a darker purple, and you can kind of appreciate the outline of this spindle structure. And then if you go over here, this is a pregnant uterine lining or muscle, smooth muscle, and you can see that the nuclei is surrounded by more of a enlarged cell type, that has a structure that kind of looks like this instead of that. Does that make sense? So that's what you're kind of trying to appreciate histologically between these two pictures. This one looks more like an enlarged hypertrophic cell, and these ones are kind of narrow and spindly under non-pregnancy state. So this is not a pathological condition. This is normal physiologic hypertrophy. Also what happens during pregnancy is physiologic hyperplasia, where you get an increase in cell number. Okay, so this is a transection through the uterus at autopsy of an individual that was an early term pregnancy. And so the darker tissue that you see in comparison to the lighter tissue more on the left, this darker tissue is because there's a larger number or a higher density of cells that are present in that uterine lining. This is where you had implantation, okay? Metaplasia, back to Barrett, our term of metaplasia, this is reversible cell replacement. So in the patient that we're talking about where there's goblet transition, if they take care of their GERD, their gastroesophageal reflux disease, then presumably what will happen is the goblet epithelium will fade, and on the next generation of new cell turnover from the stem cell layer, right, the progenitor layer, it'll give rise to normal stratified squamous epithelium like it's supposed to be there. So metaplasia is not cancer, right? It's early differentiation that moves to a new form, but if you remove the stimulus or the injury, it'll go back to its normal state. But you kind of appreciate what's happening here. We've got basement membrane, normal columnar epithelium, and then here you can see this transition of squamous metaplasia that's happening at the level of, what tissue do you think this is? It's not esophageal. It's another tube that runs parallel. Yeah, this is in the trachea or the bronchial tree. Okay, so what you're seeing here is what happens with smoking, right? You get metaplasia that happens with inhaling agents or pollutants in tobacco smoke. One second, and I'll get to you in just a second. So you've got your columnar epithelium that's ciliated. You guys remember the ciliated epithelium that lines the trachea? And then over on the right, it's squamous. Why does it go from ciliated to squamous? Why squamous is the change? What is it trying to do? It's trying to protect itself. It's trying to protect the epithelium because in smokers, chronic smokers, there's a lot of toxins that are coming into the epithelium, and so it reverts to something that's going to tackle abrasive pollutants better than something that is going to be handling, normal clean air. And so down low, and I'll get to your question, down low you can kind of appreciate, this is the cartoon drawing up top. Here's the delineation of this line, and you can appreciate, again, hematoxylin, eosin stain section. You can appreciate the cilia and what they really look like under a microscope, and you can appreciate the columnar epithelium that's here. And then here's the transition where, bless you, these cells are now flattened. You see how they're squamous? They're like flattened down. They look like this now instead of these nice columns because they've gone through metaplasia. You guys with me? Question?
So the progenitor cell isn't permanently mutating, rather than it's responding to stimuli with different gene activation to develop a different type of cell.
That is correct.
That's why it's reversible?
That's correct. So the comment that was made is accurate. Just repeat for the back of the room. The progenitor cell is not mutating permanently. It's responding to the stimulus or the injury, and it's changing. But it's not mutated yet. If it continues, could it mutate permanently? Yes. And then that's when you have a cancer situation. Okay, so let's take a peek at some of this histology in a little bit greater detail. And so you can see from this view, this is a human lung, species human, organ lung. This is a patient that had emphysema. And this is the digital histology slide, so you can kind of appreciate this yellow box here in the tissue where we're blowing up. This is a bronchus. We're going to zoom in a little bit, higher magnification. And again, we're going to look at the red and the green arrows. And the green arrow is pointing to columnar morphology that's normal, like what we saw in that first cartoon drawing. And then the red is showing where it's transitioned into stratified squamous, right, where these guys are flattening out now. Here's one right here that's flattened out. So you're getting this flattening that's taking place where it's really supposed to be columnar in appearance. And so a smoker that has emphysema, if you took a biopsy sample and you saw this, you would label this as a cellular metaplasia that's taking place. Okay, questions?
So does that mean emphysema is reversible?
So in this particular case, this patient that's wrestling with emphysema, if this is the only thing that it looked like and they stopped smoking, it's very possible that it would heal and they would get better. If you looked at other parts of the tissue and there were lesions that were cancerous, then it's too far gone. I mean, I'm not a clinician, but you would never just make the diagnosis from one sample. Does that make sense?
So the metaplasia, it doesn't always occur like on a border that's defined. It can be like spontaneous in different regions.
Correct. Yep. It can move, kind of punctate throughout the tissue. It can hit entire regions of the tissue. There could be a front that it sort of sweeps through. We're gonna get a bunch of beats here in a second. It sounds like some Amber Alert I just heard, or flash flood warning. Maybe that's what it is.
Yeah.
I didn't hear it from any of you, so maybe you did silence your phones. Thank you. Other questions on this? Okay, perfect. So now let's talk a little bit more about how the different types of injuries manifest themselves. So this is not a comprehensive list, but it's fairly detailed. These are things that can cause injury to cells. We know that chemicals, like the third bullet, you could put, for example, hydrochloric acid, right? And if you haven't thought about Barrett's esophagus, some people would say, well, that's kind of crazy. Who's drinking HCl and who's playing with hydrochloric acid? Of course, why would you ever do that? Well, we just gave a physiologic example in a pathologic state where you've got hydrochloric acid that can cause damage to your tissue, okay? Both at the level of the small intestine, the stomach, or the esophagus. So that's a perfect example you could write in that blank of chemicals to make you remember what kind of chemicals are we talking about. These could be chemicals that are in your food. These could be chemicals that are in your water. These could be chemicals that are in the air that you breathe, right? Hypoxia. Hypoxia is the lack of oxygen into a tissue. Kind of like what happens with that cardiovascular heart disease that we talked about. Then we're going to talk about hyperoxia, too much oxygen species, which are called free radicals or reactive oxygen species. We'll talk about that this semester. We'll look at infectious agents, whether they're bacteria or viruses. Those can cause pathologies, clearly. Okay, if a bacterial agent infects a tissue and now it stimulates inflammation in that tissue, that can cause trauma to the progenitor cells. Immunological reactions, genetic defects, nutritional imbalances, like iron deficiencies, okay? Physical agents like trauma, car accident, okay, sporting accident. Those are physical agents. And then, I want to make sure you guys heard this earlier, age is an absolute pathology. It's a disease. And so if you got a physician that practices geriatric medicine, they'll tell you that they handle their patients very differently, and they think about how to handle their patients very differently than pediatric physicians. Because in peds, you have a very young tissue and a young system. And in geriatric medicine, it's old, and it's under a disease state. And there's all sorts of underlying conditions that are happening in a geriatric situation or a geriatric patient. we're going to start to introduce this idea of reversible versus irreversible injury. And I'm trying to lead up to these terms of necrosis and apoptosis. This is probably what we'll pick up in about a week and a half when we come back. But I would like for you to finish out reviewing these slides, because this is where we'll pick up and we'll finish this lecture on Monday the 9th. But really what we're looking at is how cells respond to different types of injuries. And they're either going to go down a decay process known as necrosis, or they're going to go down a pathway which is called programmed cell death. And programmed cell death or apoptosis doesn't just happen in utero. You can still signal it today. And if there are enough signals of damage, then the cells in that tissue may decide, you know what we're going to do is we're going to go through an organized move out. Why does it do that? Well, in apoptosis, it can start compartmentalizing the DNA. It can compartmentalize mitochondria, cellular organelles that could be recycled. Like another cell could phagocytose it and reuse those parts. So programmed cell death or apoptosis is actually a benefit to the organism because you kind of recycle the parts of the cell that are still usable. Whereas necrosis is like a decay process where stuff's damaged and it's not usable. It's like the car got totaled versus you could reuse the engine, or you could reuse, the nice wheels, or maybe the tires are fine, maybe most of the engine compartment's okay, but it's cosmetic trauma to the body. So in that situation, apoptosis is let's save the stuff that can be reused. So we see a normal cell here. We see cell injury that's reversible, where we've got some things that are taking place, like we start organizing the DNA, clumping it. We start swelling certain organelles, like the endoplasmic reticulum and the mitochondria. The ER is important for making proteins that are important for the cell. The mitochondria, what does the mitochondria do? It's the powerhouse. It makes the energy. So it's like the engine. Like if you can recycle the mitochondria, you would totally want to do that because it's a very important part of the cell. It can be reused somewhere else. Okay? Now, the recovery in this panel here of reversible, you start seeing it reorganized and looking more like a normal cell. Whereas if it goes on and is irreversible or non-reversible, you start seeing the cell break down in different ways. If it's apoptosis, you see blebbing. You see what are called apoptotic bodies that pouch off and then dump their contents, okay? Or if it's necrosis, you see the loss of integrity of the cell membrane. It gets holes in it. It literally starts to fall apart or decay. And when the cell membrane falls apart or decay, then you've lost the entire viability of the cell, okay? So this is where we're going to pick up right here on cellular swelling. I just want to point out, we'll start here, I'll put a little note. I want to show you that there's a really nice comparison table, a couple of slides forward, that I'd like you to look at, because I want you to be able to compare and contrast apoptosis and necrosis. And this picture is something you should become prepared to kind of discuss. This scanning electron micrograph showing apoptosis in one picture and necrosis in the other. So we will back up and we'll start here, but I want you to kind of look forward and kind of finish out the rest of the slides so you can come prepared for Monday the 9th, okay? So you guys have a great fall break. Hope you get a refresher, because this was a stressful week for you. Thanks for your attention.